Hi there, I'm back with another JBrowse tutorial. This tutorial will explore how to use the new faceted track selector plugin in JBrowse browser. It's a new way to filter tracks for which ones you'd like to select for viewing in the JBrowse browser. And in doing so, I will show you how to use that while exploring the new set of NGS data tracks, the next-gen sequencing data tracks that we've retrieved and imported from the Gene Expression Omnibus database. So that's the GEO database. So we've gotten a large set of tracks from there, approximately 500 tracks so far, spread across Lavis and Tropicalis, the latest genome versions. So let's go exploring. Let's go to genomes and let's go to the Lavis 9.2. genome version on JBrowse. Okay, so you'll see that the new set of GEO tracks have all been added here in the hierarchical track list, organized into three primary hierarchical categories: ChIP-seq data, RNA-seq data, and manipulations. So these are further sorted and ordered into subcategories according to tissues, as well as stage, and sorted as well accordingly by tissue and stage, as well as other attributes like methylation and transcription factors and other attributes of the actual GEO data set from which the experiment was performed, from which experiment was taken from. So as always, you can select these tracks as normal from the hierarchical track list, these GEO tracks. And if I hadn't mentioned it before, you can always also filter the track list down to only tracks containing a certain keyword. So if you're looking for tracks related to foregut, for example, it'll only weed the track list down into tracks that have the keyword foregut in the track name. So a lot of totals will go down to 0 in these categories, but it'll only show the categories left that have foregut in the name. So let's just X that filter out so we see the full track list again. Another thing you'll notice with the GEO data tracks is we've also added not just bigwig data tracks, but for some bigwig experiments, for some bigwig data tracks, there are also corresponding peak call tracks that relate to that same experiment performed in the bigwig track. So if you click on two of those matching tracks, you can see the peak calls. The peak tracks are based on peak call files retrieved from those GEO experiments, and that basically we've just shown the track as blocks where the peaks would occur, and so how they relate to the signal of the bigwig data tracks in the corresponding experiment. So let's just close those tracks now. So you'll see there's some peak call tracks for Lavis. There's more for Tropicalis, but not for every single bigwig data track, only for the ones that were available that we could process. Another way, so the new way, if you like to use for filtering which tracks you'd like to view in the JBrowse browser, is the new faceted track selector in JBrowse. So to access this, you just click on this tab here, labeled GEO tracks, and it'll open this faceted track selector table. Now this shows not just the track names for each of the bigwig GEO data tracks, but also other attributes of the metadata of the experiments that we thought were informative, like assay, stage, tissue, what lab produced the GEO experiments, publication if one is available, the GSE number of the GEO series of the whole experiment, and the corresponding sample numbers and the GSMs. These are only the bigwig data tracks from these GEO experiments, not the peak call tracks. So those peak call tracks you select only in the hierarchical track list as before. Now this will allow you to select tracks based on these metadata attributes. It also allow you a few helpful links. So these publication links will go directly, if you click on one, to the Xenbase publication page of that paper from the GEO experiment. Close that. As well as this link will relate to the GSE ID of the GEO experiment. If you click on this, let's say for GSE 76991, it will go directly to that GSE page, that GEO series page on Xenbase, which provides you even more information about the GEO data set. You can view differential expression data and more information about each sample. It also has direct links to the article again that relates to the GEO series. The GEO series page directly on NCBI, so the original source, and a link to our Xenbase FDP, which will go directly to that folder, which contains all the data for this GEO series. So all the underlying data files, like the bigwig data files, peak call files, if that applies, as well as differential expression analysis and other background data files related to the GEO series. Let's close that. So aside from those links, again, it gives you a way to select which tracks you'd like to see viewed in the JBrowse browser, but based on all this other metadata attributes related to the GEO experiments. So you can do this just by looking at it and clicking which ones you'd like to see in the JBrowse browser. Let's say if you select one or two here, go back, they'll show up just like you would if you were selecting them from the hierarchical track list. Now if we go back here, deselect them, you can also, the main power I think is to be able to search from this text field here for keywords not just in the track name column, but in any column of this metadata. So again, if you're looking for tracks related to foregut, it'll search not just in the track name, but in the tissue column and every other column that you see here related to the GEO tracks. So again, let's try selecting a couple of these tracks. Let's clear our filters. And you can see those two tracks have been selected in the JBrowse browser. If we go back to the faceted track table, again, if you always want to see if you made a couple of selections after a few different searches and you want to see which ones are currently selected, maybe it's a bigger list than this in your scenario, then you can see right here. And then, of course, deselect all the ones that you currently selected before. Go back to the original table, back to the browser, and they'll be deselected. So that's how you can play around with that faceted track selection. And it works, it harmonizes, it works in tandem with the hierarchical track list too. So whatever you select there will be selected in these checkboxes here too. So this faceted track selector and everything you see here, the way the tracks are sorted and ordered, it's identical basically for the Tropicalis 9.1 genome as well. So just a different set of GEO tracks available there, but all the functionality that you see is available there as well. So that's how you can explore the GEO tracks and use the faceted track selector.